For joining us tonight. Thank you, Julia. So everyone, welcome to Science Cafe. We are so excited to be back for 2021. And my name is Allison. I'm a PhD student in the biology department at UMass Amherst, and I will be facilitating today's conversation. Some housekeeping things. Life Science Cafe was established in 2011 and is organized by graduate student researchers at UMass Amherst. Each month we invite different experts to discuss their research, and we typically host these events in the Amherst community in person, but are happy to be able to continue bringing this information to the community over Zoom. So with that, we would like to thank our sponsors. Let's see, we have the UMass graduate program, Organismic and Evolutionary Biology, the Graduate Student Senate, and the Graduate School at UMass, the National Science Foundation, and our generous UMass Gives donors. For our next event, we will be discussing how enzymes work with Dr. Rivera Colon on March 11th, so mark your calendars. And in the chat, if not now, hopefully by the end of the conversation today, there will be a virtual sign-in sheet. So please take a second at some point this evening to fill that out so we can get a better idea of who our audience is now that we have shifted online. So without further ado, we are very excited to have Dr. Li-Jun Ma here with us today to talk about fungal diseases in plants. Dr. Ma received her bachelor's and master's from Wuhan University in China, then her PhD at the State University of New York, and then did a postdoc at Harvard. She then worked as a researcher at the Broad Institute at MIT and Harvard before joining the faculty here at UMass in 2011. She is a fungal biologist, genomicist, and plant doctor, and we are so excited to have her here at Science Cafe tonight. So before we get started, we're going to start with a little poll to see what our audience knows about fungi. So let me see if I can. Okay, so if Julia could start the poll, please choose which out of these beautiful organisms is not a fungus. And we'll give everybody a couple minutes. All right, we've got almost all the votes in. If you haven't voted yet, get your vote in. I know it's tricky. All right, we've gotten almost everybody in. And let's see, here we go. Can everybody see the results? We can see that indeed we have picked a sufficiently difficult question because G is the incorrect answer. This beautiful purple thing is indeed a fungus. And the correct answer is E, which is one of, I think it is the world's largest flower of the stinking corpse lily. So it's pretty wild to see all of these different types of fungi and mushrooms. And so we thought this would be a good place to start. I will go ahead and stop sharing. We thought this would be a fun place to start now that we've kind of seen some of the wild and beautiful diversity of what are fungi. Can you, Dr. Ma, give us kind of an introduction about what is fungi? How can it possibly make all these very, very different looking things? And like, where do they live and what are they? Can you give us a brief background?
Great. Well, first of all, thank you so much, Alison, for that generous introduction. Actually, it's true. So my starting in UMass, it's the same time as a cafe started. So yes, I heard about
Yeah.
long time, yes. And I was invited to come here some time back. And I'm so glad it happens today. So very excited. I think the story about fungi sometimes is underappreciated. So that's why that beautiful mushroom was not recognized. So to help the audience, I think I get a little introduction about fungi, maybe very quick, just one slide. So I'm going to share briefly, very quickly. So fungi very diverse and they are wonderful. So I actually just finished this puzzle this week. So this is a game given to me as a holiday gift from my wonderful daughter and her wonderful boyfriend. And it took me 2 months to finish it. But you can tell is that there are so many mushrooms we all know. But one thing maybe we didn't appreciate that is that we need them. We're not only eat them, we need them. So they are pretty much a recycling factory for the planet. So when you're hiking in the mountain or in a trail, if you see a mushroom, so they are doing a very important job is to turn this debris into usable compounds for the plant or whatever needed to use it again. So it's very important component. So every time when you're walking on the wood, sometimes it looks dark and dirty, but they are still doing their job. And the other aspect about fungi we probably all know, which is many of our food are involved in the fungi. So here the cheese are made out of mushroom, don't mention. So cheese, the truffle, and also the yeast we use making beer, making maize bread. So that's all very important part of our day-to-day life. So one thing people may not know as much is actually fungi are also very important sources for medicines. So here it's, I don't know, in the audience, maybe it's a quick test. It's, do you know what is this compound for? What this fungus is? And so this is producing the first antibiotics. Penicillin is coming out of a fungus called penicillium. And even today, we still go into the fungal kingdom and to ask them to say that, now we have a disease, we don't have anything. Do you have some tricks we can borrow? So we kind of still learn from them from that aspect. So that's kind of general sense to the introduction about fungi in general.
Awesome. That
Okay.
is great. Yeah,
Good.
they are, I guess, everywhere, and we really need them for a lot of different things.
Absolutely.
So I guess one kind of basic question is, I guess, about what fungi are. Fungi are is
Mhm.
like, we know plants, they photosynthesize, and
Mhm.
that's how they get their energy. And we know animals get their energy from eating
Mhm.
other things. So how do fungi really get their energy?
Good question. How do fungi get their energy? They do not do photosynthesis, so they take from whatever. For instance, if they hop on a wood, so they degrade the wood and they eat it. And if they hop on human and they eat the human, so that's kind of say they taking the nutrient from the environment using the degrading enzymes.
Okay, cool. So they're kind of like stealth predators.
Sort of. I think the most time in the benign ways. Yeah.
Yeah. Okay, so where exactly in relation to plants and animals do fungi fall if we're talking about like the tree of life? Because I've heard that we are more related to fungi than we are to plants. So can you kind of give us a little bit of a kind of orientation to the tree of life and where we all kind of fall?
Yeah, so okay, so this is one of the image. This one was published in 2012 by Harvard Hughes Medical Institute, basically putting the whole Earth into one linear line. And then so so here where the Earth's activity starts, and here where we are today. So sort of like that. If we're thinking about sort of the history into 1 year frame, we will have 4 quarters. And then if now today is the close to the end of the year holiday season, and then we know that animals arrive around like the end of October, November sort of ish, definitely around that time. And where fungi comes in, actually not too far, not too much earlier than animals. So they probably at the end of October, beginning of November, and really kind of close with animals. And where plants come in, actually much earlier than both fungi and animal. So they are, say, definitely in the season of after the summer, early fall-ish. What it tells you that is actually fungi arrived or evolved into the planet when there are plenty of plants existing. And that related to your question is where fungi get their food, right? So before they successfully establish their life and their life cycles, and they are already photosynthesis organism very established in the planet. So fungi have very intimate relationship with plants. That's why. So I want to show a few images. So this is one of the symbiotic relationship, earliest the single cell algae and together with fungi forming this lichen structure. We see this in the tree all the time. So lichen is very important establishment in a lot of ecosystems. And what happens is you will see that this one, the kind of fiber structure, it's a fungal hyphae, and this singular one are the algae cells, and the algae has a photosynthesis capacity, and so therefore they do provide the nutrients to the fungi. So this is in the early stage between the early symbiotic relationship between fungi and plants. And but then the question is, no one like just take food from me. That will be pathogenic relationship. So most fungal plant interaction actually is a symbiotic. So what fungi provided to this the cell is nutrients like nitrogen, phosphate. So they give each other important things. Therefore, they can survive in very tough environment. So here are the two kind of kingdoms lying side by side. We'll see that so fungal kingdom arrive in the planet. Plants are very successful already. But however, the establishment of the fungi played very important role for the plant kingdom as well. So here it's a line to connecting the land plant established. So when plants started, they are aquatic organisms, so they need water to survive. So go from the aquatic environment into the dry land is a huge evolution event. And if you're looking at the alignment of these two kingdoms, when the land plants established, and they are diverse fungal species existed. No question about that is the success of the origin of land plants are heavily depending on fungi. In which ways, right? So if if we're looking at this image, what we see is a mushroom on the ground. What you don't see is underneath this mushroom's hyphae are forming mycorrhizal relationship with plants. So I thinking this number is arbitrary, but almost all the plants need some kind of mycorrhizal relationship to be successful. So this is one of the example. If you see that's very tiny pine seedlings, the bottom of all this white hyphae are interact with the root. So the structure is bigger than what you see on the surface. So basically, so they are benefit from each other. So most time it's very symbiotic, one support the other sort of relationship.
Wow, that is pretty incredible, that the fungi enabled the plants to kind of move on to land. So in this, in these two trees that we're looking at, or the phylogenetic trees, the above is the fungi, correct? And it's kind of like expanding right at the same time as the plants have moved onto land, right? And the plants are the lower one.
Right, plants are lower one.
Okay.
So you can see
Wow.
before this, there are plenty of plants exist. It's just this is origin of the land plants.
Wow. Well, that is fascinating. So you mentioned that most of fungi plant interactions are beneficial, in that they both give and receive something, and allow them to live in new areas. But you've also mentioned that fungi can be pathogenic. So in what cases are fungi pathogenic, and why did that start happening, as opposed to this kind of mutualistic relationship that was happening? What does the what led to that kind of exploitation of the plant?
Yeah. So imagine when the fungal kingdom involved, the plant kingdom already there. So the interaction start from day one. They have a talking for the give and the takes. So that's always existing there. So we already know that both plants and the fungi have very successful immunity, just like us. So they will be able to recognize this is something happening. And do I know you? What do you bring to me? What I give to you? So this interaction is sophisticated. So they are molecular mechanism to dissecting. So this part of the research we're doing, try to really understand how do they talk to each other? Like who is knocking the door, right? Who is passing the food, and what is the return? But that happens. But when this interaction balance broke down, once it's broke down, disease happens. So I think I have a couple examples to show, plant causing problems. So the reason it does cause a problem is the balance broke down. And so I think one common example, probably if we have a listener from Amherst community, you probably see some of them. So this is when you're growing your tomato, you just get wilted it out. Before you even can harvest anything, plants cannot dying out. And I want to show another example, which is pretty devastating. So that why not eating the bananas your grandpa eating? So if you go to grocery store, like 90% of the time, the Cavendish banana is what you see in the shelf, and pretty much you get used to eat it. And this banana is not the same as the banana we have it 50 or 60 years ago. And so I think some white bananas still resemble this banana. It tastes more fruity and it's delicious. And it's not because I think they are not good, so we don't eat it. The problem we don't eat the same banana is we don't have it anymore. So the history was like that around the 1950s on the old banana. So we call them the Gros Michel. So Gros Michel suddenly start to die out. So it's so devastating. So the whole field just goes away. And disease was reported, identified in the country Panama. So it got a very bad name called Panama disease. And the severity of the disease really is wherever it reaches, it kills everything. So therefore, whole industry got very nervous, and because it's very important input and export business. So plant biologists did step in and said, okay, so this is the disease, and people start to know it's caused by a fungus, and say, how can we deal with it? Around the 1950s, so the most successful efforts was going through the plant breeding efforts. So they identify Cavendish banana, and it does resist to this disease. So bingo, so it took over. And the success itself inherited some problem because it's so successful, and it started growing all over the global. So when you only have one food, you're kind of causing problem already. So the story, so here is where actually our research start to connect it. So around the 1990s and in East Asia, people reported this banana start to die out. And the disease symptoms is very similar as a Panama disease in the 1950s. But community and society does not want to believe that, because Cavendish resistant to this disease shouldn't be the problem. So people didn't have a very successful diagnosis. The disease spread from the East Asian and into China, and then around 2012, it reached Africa. So the situation was devastating. Around that time, there's a lot of talks about we're going to lose our banana. Of course, scientists doesn't want to give up for that, but it could cause devastating problem. The reason that, as you're asking, why it start to be problem, so it is the establishment of a fungus. It had been very successfully break down the plant immunity. And because the monoculture practice with the banana industry, you only have one type of banana. If one pathogen had been established the way to overcome your immunity, you just lost control of the situation.
Wow, so it sounds like it's particularly problematic for agricultural
Absolutely.
plants, because we, have these single monocultures of all the same genotype that have the same level of immunity and are then so susceptible to it. So at this point, I think it'll be good for us to open up for questions from the audience. We'll maybe take 1 or 2 or 3 questions. If you have a question, go ahead and put it in the Q&A at the bottom. You can type it in, or you can also click raise hand, and then you can speak your question out. So, all right. We have our first question. We are told that infectious organisms which kill their hosts tend to die off because that's maladaptive. Do we know why that did not happen with the Gros-Michel fungal disease?
Such a beautiful question. Yes, so general, because as we said, most relationship is symbiotic. And so this one kind of continue with the killing, where they're going to end it with. So the development, I didn't talk about that, is the Gros-Michel pathogen, even though it's the same fungus, it already evolved drastically. So maybe I'm going to talk about this particular fungus. How could it be so powerful, so successful? How did it maintain the evolution forces even under all different kinds of selection pressures? So I think we probably will touch on that once I go into the way. So my lab have been really using genomics approaches. So then we really could open up the genome, looking at every single base, try to understand what they do. And evolution forces driving the evolution of this particular group. It is one of the working focus of the group. I guess I will save that for a little later.
Okay,
Remind me if I didn't answer your question later. It's probably a bit easier that way.
that sounds good. All right, we have one raised hand from Sarah, so I will click unmute. So Sarah, you will be able to ask your question out loud.
Hi.
Hi, Sarah.
I wondered if this is a similar situation as they had in Ireland during the potato famine. Was that a fungal disease as well, do you know?
So potato famine is a fungal related disease, but they are equally bad. And they also use very similar machinery to kill the plant. And same thing, that pathogen are able to overcome plant immunity and start the killing. And it's also because they overcome the plant defense system. So it's also sort of, at that time, I think they have one particular potato sort of thing.
Yeah.
Always diversity is the key to overcome the situation. But that one coming from something called Oomycete, it's not belong to the same kingdom, but they are very similar. They have a lot of similarities, Sarah.
Great, thank you.
Sure, you're welcome.
All right. Oh man, we have so many questions already, but I think we are going to keep going on to our next section, and keep your questions. We'll have them in the chat and we can try to address them in the next break for questions. So now that you've kind of alluded to how your lab is involved with studying all this, could you give us I guess a brief explanation of how exactly do you study this, and what is the aims of your group to try to help figure out how to protect these plants from these diseases?
Okay, so maybe we'll touch base on that evolution sort of the question. Move on to this one. So even though when Allison do the introduction, call me plant doctors, I thought that's far away from who I am, but I really inspired to be. So one inspiration story was actually really coming from a friend. So this friend, artist from South Africa, and when this disease reached South Africa, so artists come to visit this farm. When he called me, he was really in tears. And what that means is this is a family farm. So the brothers, sisters, parents, everyone put their money into this little farm. When the disease hit, and they had nothing. And because the disease is so severe, it's very hard to see what's the kind of, you can manage that. So one thing we realize, the disease spread have one reason. When this problem comes, the resistance to say this is caused by particular pathogen. Because if you don't know what's the cause, then the management is less. So we do thinking that find a way to know who is the cause of the problem is essentially important. So this is, so I think I went with all this to one of the diseased field, and that's what we start to see the phenotype of the plants. If you cut the stem, you will see this kind of black spot. That's indication of this fungus, maybe it's there. And so here's another, this is not from banana, this is from a basal stem, it's infected by the same fungus. And then you can see their structure, they have their fungal, really fungal-like structure, and then you have the spores. So in my lab, we will do more fancy methods and do different kind of fluorescent protein to tag it, we'll see its behavior. So the whole disease we already know, which is a model system in my lab to work on, is called Fusarium, oxysporum. And so that's coming back to the evolutional questions. So, okay, so the Fusarium oxysporum is responsible for the Panama disease. And Fusarium oxysporum is also responsible for this recent outbreak. So we call them TR4. So what's the difference between TR4 and 1950s disease phenotypes? And we also know that, what's responsible for the tomato disease as what I showed it. So then I thinking this model system coming to be very intriguing for us. So here we will go into that. We call Fusarium oxysporum a kind of species complex. And within that species complex, we know that it causing disease more than 100 different plants. So here we show banana, we show tomato, many of other plants all have this problem. So when I start to kind of establish my lab, I realized one extraordinary about this fungal system is that they're not only causing disease on all those different plant species, they maintain host specificity, which means if I take the fungal strain from banana, it does not cause problem in tomato, vice versa. So the really intriguing question is, how could a fungal system do such a job? So that was, for me, in 2010 or 2009, it was an unanswered question. So, and we decided to take in genomics approach, just sequence it. So that's the work published in 2010. I think it is a surprising discovery. So when we're looking for the different genomes, we find that they have all those bars. Each bar represents a single chromosome. So if you have any pathogen coming from same plant, species complex, Fusarium oxysporum, they more or less have a bar of like that. It's the same. But then we have another second part of the genome. We call them accessory chromosomes. And this accessory chromosomes only special for one particular pathotype. For instance, if we're looking for the tomato pathogen, they have 4 unique for the tomato pathogen. If we're looking for banana pathogen, they have a completely different, but they also have this kind of structure separate. So it kind of making sense is that if each one of them maintain this core part that define who they are in general, they belong to. But if they carry the different weaponry that will enable them to be enemy to different plant hosts. So specifically, they just overcome different host immunity for them to successfully establish their interactions. So this story was started, we have this idea, but how real it is. So this work was done with a brilliant graduate student, PhD student from the Netherlands, Amsterdam University. And I remember when I visited them, I really love that. We go to a coffee, we sit there and we just chat. If that's true, what could be, what it look like? And then she is brave. She said, okay, if that's true, then if I can put this machinery, push it to a different fungal strain, can it cause disease? So I think at that time, we do have the system. We know this one, it causing tomato. It basically killed the tomato. And then it does have another strain, and we know phylogenetically the two are very similar, which means if you're looking at the core are very same. So then she said, if I can forcefully put this chromosome into this well-behaved fungus, what it look like? So then she did that. And out of the surprise, or pleasant surprise actually, once she can prove this chromosome successfully landed in this well-behaved fungus, it start to kill the tomato plants. So actually, we not only kind of did a sequencing, did a serologic analysis, we also have evidence to prove that if you can carry that weaponry and you can kill the plant host. So that kind of gave a certain conclusion about the idea.
Wow. Okay, that was so amazing and so that was so much. So I kind of want to, if that's okay, can we go back to the last slide and kind of walk through one more, back to where you have the chromosomes on the slide? Yeah. So yeah, this is just mind-blowing to me that one single species can differ so much in the genes that it has. So I guess we're used to thinking about genes and genomes as like, we are 99% or 98% similar to chimpanzees, or, like this is so different that entire chromosomes are different within a single species. And so I guess I'm wondering, like, what is this common in fungi? Is this common in plants to have this much difference? And I guess if you could put a number on it, like how different would you say these strains of this fungal species can become genetically?
So the phenomena of this kind of accessory chromosome concept did report it more than in the fungal kingdom. So they are, for plant, if you're thinking about B chromosomes, you probably heard about that. And in fungi, their connection of the presence of these actual chromosomes with their variants probably is the best studied cases. This the more than Fusarium genome contain this phenomena. Other fungal species also have that. So the ever asking question will say, where does chromosome come from? How could they migrate from one to the other? How to dictating that? It's not solely understand. There are many interesting features here. So for instance, I put them to different colors, right? And there are good reasons to put into different colors. The gene contents here are very different. The structure contents also very different. For almost all this kind of accessory chromosomes, they have tons of junk DNA there. But junk DNA, does it just the junk? May not be true. So I thinking kind of connected to the previous question, one of the people was asking is that what's the evolution trajectory of this organism, right? So we also very curious. So the lab did experimental evolution. Just, okay, so now you have the genome, right? So we know works fine, you can kill the host. Let's keep you in the lab. So we try to keep them in the lab into 3 different conditions. One is to spoil them, right? Give them sugar, give them fat, give them whatever make them grow fast. And the other them say, well, you still have to kill plants. So we passage them through the plant host. The other one give them little stress conditions. So it was a surprise, half a year in the lab, half a year, 6 months. And what the condition is, if you passage the same fungus, started the same thing to this kind of spoiled condition, very comfortable, very rich, you just keep growing. They start losing this stuff. But if you keep them go through the plant host, they keep it. So which means what happens is we find that if we rate the evolution process and this actual chromosomes have highest rate of cumulative mutations. So they kind of provided a hotspot to changes, and accumulate new genetic material, and enable them to adapting to new stresses, whatever they may encounter. So answer the previous question, that is, we don't believe the system is a dead end evolution, even though we did this very recently. So one of the MCB students just finished one set of experiment. We just heard the talk this week. So which means we are still on the early investigation of the whole thing, but it's intriguing what's happening. Did I answer your question or did I go too far?
My question?
Yeah.
I think so. Yeah. No, that was.
Yeah, you was asking chimpanzee and human. But I guess my point is that you cannot just looking at the statistics. It is key is what are the genes are changing and they perform what kind of function.
Wow, well, that just blew my mind. So at this point, I think it'll be another good point to break for questions. And I think there was one question from the previous set that I'll ask first, and then everybody feel free to put more questions in the Q&A. So the first question we have is, how does the fungal immune system work, and how does it kill invasive organisms?
Fantastic question. And I will say that we know a lot about human immunity, so that's very well studied. And the plant immunity was exploring in the recent years, like in the past 20 years. Fungal immunity, just the baby step. So very baby. So because if you imagine fungi are living in an open space, they need to defend themselves against the virus, against the bacteria, and need to interact with the plants. But because, as I said, the plant-fungal interaction has been the center for the evolution process. So there are more studies right at the fungal-plant interface. So like our group also doing some small amount of work. What we know that is, so both plant, fungi, whatever, they are individuals, and then they have their characteristics. For plants to recognize the presence of the fungi, or for the fungi to recognize the presence of the plants, at first they need something in their cell walls, cell plus membrane. So receptor definitely play a very important role to sensing what is present in the environment. And once the sensing started, then they need to reprogram, say, okay, now we need to do differently because environment changed. So for fungi, as you was asking me, how do fungi get food? So they do observe the nutrients from the environment. So they are very powerful to sending enzyme out, sequester the nutrients out. And that is also part of their negotiation tool with the plant host. There are so many different ways they are doing it. So one way, I think my postdoc really loved that story, is that fungi very smart. So they said, okay, I want to protect myself, so I will hide in myself. So they produce the protein when they need it, just cover their wall. So then say, nobody see me anymore. But the true answer is the detailed, like, understanding as a human immunity level, we're way behind. So a lot of molecular understanding, it just start to open up. But the truth is, yes, if fungi sees bacteria, they react differently. If they see virus, they react differently. They definitely are very good at reacting to the presence of plants.
Awesome. Great. Thank you. All right. Our next question is, do endophytic Fusarium oxysporum, I don't know if I pronounced that correctly, but the species you were just talking about, do these endophytic strains possess the same virulence and pathogenicity genes as pathogenic strains? Okay, that was a lot of jargon in that question. So if you don't mind, yeah. So if you don't mind breaking that down for us of, what is endophytic and what is pathogenicity and virulence, some of those kind of jargony terms, if you could explain those as you answer this this complicated question for us. Thank you.
That's perfect. Okay, so I did prepare this slide. So, okay, so yes, the question have so many layers. So Fusarium oxysporum, when I introduce this one, is that it killed tomatoes, it killed bananas, but it's not always bad. So here is one of the example is, we know the fungus are coexist with the plant. This is a plant root, and so the blue color indicating the fungus present there. And actually, one of my graduate students did a very careful job is this one actually make plant grow a little faster. This is the model plant, Arabidopsis, we play in the lab. So it's kind of, it's toy. And then, so the endophytic is that it coexists with the plant, but does not kill it. And so we just actually, I don't know who asked this question, we just have a paper about to submit, which is comparing this endophytic interaction versus pathogenic interaction. So pathogenic is easy to understand, kills the plant. So here is how does it do the killing is the fungus enter the center of the plant. So you see the blue is all over the place. And then the first phenotype is that once it's occupied the xylem where plant taking the water from the soil, if you block it, plant is dried out. So that's the wilting structure. So go back. So, okay, now we have two systems. We know even they belong to the same fungal species, but they could cause very different results. So one we call them endophyte, the other one we call it pathogens. So we know the system exists. We also did a genome sequencing. I think I will be with this one to asking the question. My initial hypothesis is, well, if it does not causing disease, it probably does not have the actual chromosome. But you can see how many times we are wrong. So when the data comes in, that's not true. So even this endophytic strain contain a complete chromosome. But the genes within this chromosome is completely different from the gene from the pathogen. And so we did a careful study about this work is how do they do that? How do they turn this interaction? So we know they all interact with the same host now because they all Arabidopsis. And then we know they all have actual chromosome in addition to their core. So how the interaction happens. So the story is long, but the short answer is that overall, Arabidopsis recognize they are Fusarium, regardless they are pathogen or not. That was intriguing. So we put them together, then we see all the genes involved in the process. They say overall majority of genes are the same. But the difference is that endophyte somehow be able to negotiate with the plant. So here is one of the story. The choir dance, say, don't worry, I'm not going to kill you. In the contrast, it promoted the nitrogen signaling pathway. So even though started the same recognition, but the interaction coming to be very different. And this actual chromosome played a very important role in this kind of attenuation, promoting growth activity, contrast to the killing effect. So I think.
Wow. That's cool. So let me see if I got this right. The good version and the bad version of these strains could be very genetically similar, and they could both have these accessory chromosomes, right?
Yeah,
but
different
the genes,
chromosomes. Yeah.
Okay, but so different accessory chromosomes altogether. Yes. Okay, yeah. So, okay, yeah, that makes sense.
They are very different in the sense you was asking, how do fungal immunity working with the plants, right? So we know that the fungus need to, when we say they have machines, they have chisels, they have hammers, right? We know how these hammers, chisels look like. We know that. So that for them to kill the plants. For this endophytes, it have 0 such characters, even though it has accessory chromosomes, but it does not have the weaponry to kill the plants. So now it's a time to understand what do they have, right? How did they do the job to promote the plant growth instead of killing plants? So I think we are in the early phase of this continuous exploration.
Wow, okay, great. Thank you. So we have so many questions, but I think we're gonna go ahead with our last section, and then everyone hold on to your questions, and we'll have one final round of questions at the end. we do have a quick poll, another quick poll to give everyone. So if Dr. Ma, you don't mind stopping your screen sharing, and then I will share mine. And let's see if I can. Oops. Okay, so we have a task for the audience to please help us diagnose our sick plants. So Julia, if you could start. Okay, great. So we have plant number 1 is this one here on the far left, plant number 2, and plant number 3. Give us your best guess of what you think is making these plants sick. And don't worry, I think it's supposed to be tricky again. All right, we'll take just a few more seconds. All right, I think we have just about everyone in, so we can take a look at our results. All right, looks like we had a good turnout for the first question. A lot of people successfully guessed that this was a nutrient deficiency. Plant number 2 and 3 looks a little more confusing. And plant number 2 is a bacterial infection, the citrus canker disease, which it's okay if you didn't know that one because we haven't talked about bacteria today. And the third one is an example of a fungal infection. Not what we've been talking about so far, but leaf rust, which is another one. So congratulations everyone for successfully passing our second poll. And now we'll get back to, let's see. We'll get back to our third set of questions. So we've been talking a lot about how you hope to use this information that your lab is studying to actually be able to help reduce fungal infections in crops in the real world. So can you give us a little bit of kind of insight on what you're working on right now and kind of how you're hoping to implement this into agricultural settings, say? And feel free to also start sharing.
I think I'm just, I think I'll give it a try without it, maybe. So, okay, I do it. That would be easier. So I thinking as a scientist, I always believe in science. So therefore, I always go back to science to understand it. So when I'm looking at this image, so we know that collectively it killing so many different plants. But if you're looking at individual cases, if you're looking for the pathogen infecting banana, right? All other plants are actually winning the battle. So overall plant immunity is very strong. So the banana pathogen was outsmart the Cavendish, therefore it caused the disease. But collectively, the immunity is strong. So therefore, we really wanted to understand plant immunity from the baseline. So I thinking fundamental understanding, sometimes thinking so far away from practice, right? So you just want to do your game, you want to understand what's going on there, but I don't believe it. I think fundamental understanding will dictate the practical uses. So that's what I said. We're going to the lab, we use Arabidopsis, we really want to dissect. But in the practical sense, I told you that when my friend from Africa called me with tears, without knowing that, say, if we can diagnose, if we can do quarantine effectively, then maybe that farmer could save their life, sort of thing. So we are really kind of invested to see whether we can develop powerful diagnosis tools. So this is related with the banana research. So we know the pathogen involving from 1950s to 2000 have been changed. And if we looking at everything without knowing what is special about the 2000, the pathogen look like, and we are not powerful. So we develop something, this is we done in the lab, the primers. We will see that this set of primers only detect most recent pathogens, so they can differentiate. And still this has been done carefully in the lab. So we want to be this one, be able to patch into the field. So this is work done by one undergraduate student before COVID hit. So what happens is she will say, well, if you can see the pink color through the coloration, you know you don't have the reaction. If you see yellow, you know you have it. So here she have 4 tubes. She can positively detecting 2 out of the 4 negative. So I think in the technique, it's promising. We're still on the early phase of this development. The other thing is that this is a collaboration with friends, collaborator in Rutgers, which is really heavily dependent on plant defense. So his team identify resistant to plant cultivar and then go to the field to practice. This we visit a farmer because this work has a big impact. So UMass, together with my friend Rob Wick and Ann Gertjensen, we organized a basic workshop to introduce this. And the lab I heavily invested to understand why this one is more resistant than the others. So I thinking it's always 2 hands. As myself always understand the limitations, so we heavily depending on friends, collaborators who really work on the frontier together with farmers. That's
Awesome. Great. Well, it sounds like you all are busy. So that's really exciting to hear. So we have just a couple more questions before we wrap up. And one of our goals here at Science Cafe is to kind of bring, down the walls between scientists and the community. And so I was wondering if you could give us a little bit of your personal background and kind of your what got you into this, what got you interested in this topic, and kind of what is your kind of lab philosophy and scientific philosophy regarding topics specifically about diversity, and how does that play a role in your lab and in your work?
okay. First of all, I will say it's such an engaging conversation you had facilitated. Thank you so much. And of course, it's a huge question. Since this program, as I was highly praised for both you, for all you and everyone behind the coffee program run by graduate students, I feel I need to honor you by answering questions with my own experience. So I'm a first gen college from my family. I will have all the different hats, right? Like you said, I'm a fungal biologist, plant biologist, a plant doctor. And if I call myself the first title I have, actually, it's a mother. I always believe that's my primary job. So I want to show my kids. So I have 2 of them, beautiful. And so I always feel that is. So the reason I want to say that was I never thought my driven is to be successful professor, and rather is that I flow with life. But the curiosity about science always drive me back. So I wanted to say that I went to PhD program after my son is 4 years old. So I don't think that's something impossible, especially for the women students. I think everything is possible. And I know it's hard, I was worried, but I determined not to let worry stop me, rather let the curiosity guiding me. So I thinking that's where I was able to walk in one step at a time. And after working in UMass for 10 years now, I could confidently say it's such a rewarding position. I was so fortunate to have working with so many wonderful students. So I think the other layer to answer your question, Will, is how did I decide to working on Fusarium system? I'm always curious about science. I always view science, it's like a game. It's, the answer, it's hidden from you. If you're smart, you will be able to find the answer for. And just like any advanced game, it never ends, right? If you answer one question, the new question always derive. It's just such a attractive process for me. It's always, I see a question, I think I can find the answer. So it's just every step just linked together. So in 2010, I sort of defined this accessory chromosome structure. I just know there are so many questions. I cannot say goodbye to that one. So I feel I have to follow it. So it's basically the system asked me to start a new research lab. And I thinking right now I have a bunch of kids working with me, and we all have so much fun with the system. It's so many different questions we need to ask. And today we didn't have a chance to talk about this fungus also cause human infections. And so we also have two systems compare and try to understand what's going on. So I know it's run over time, so I better stop.
Yeah, I wish we could keep going because I know there's just so much more to uncover, but I just wanted to again say thank you so much for coming and being with us today and for sharing all of the amazing work you're doing. If you are able, we could take a couple more questions, but we understand if you also need to go. I know it's a couple minutes after 7. But
no problem. I can stay. If more questions, I'm happy to answer.
Okay, excellent. Yay. So we'll just try to squeeze a couple more questions in here. Sorry that we're going over, everyone, but this is just really exciting. Oh, one final note, if you are still around, please take a second to fill out the virtual sign-in sheet. That will definitely help us make our cafes better. Okay, so for those folks who can stick around for a couple minutes, thank you, Dr. Ma. The next question we have here is about the evolution in the lab. Does that say something about monocrops like banana? Does that help keep the genes around?
That's interesting. So the experiment we do in the lab is not directly related to banana case, but yes, so it will be indirectly directing our understanding, which is that when we passing tomato passing, we cannot grow banana here. So doesn't matter how much I love that line of research, we cannot grow banana. So we have to be practical. So what happens is we passage the tomato passaging through tomato. So we will see that the selection pressure is very strong. After half a year, we can select fixed strains. So the strain already different from the beginning and have a stronger fitness. So which means it can tell us if we allowed the passaging to happily living with its host, right? It will be a better passaging over time. That's the same practice when we say, if you have an infection, you better eat antibiotics to kill it. If you just have a killing, suppress it, if it survives with you, and will gain resistance to whatever the means you have to control it. So it's the same way. So if they are able to survive under the same host, they will be more effective. It will be stronger passaging. So that's, I think, in our evolution study, definitely provide a solid answer for that aspect. So in that sense, diversity is important, right?
Yeah, definitely. Okay, so do you have time for one more question maybe, and then we'll wrap up?
Sure.
Okay, excellent. Last question for this evening, and then for anyone else who has further questions, feel free to send them to our website, and we can try to track the answers down for you through Dr. Ma. Okay, last question. Are there other immune species of bananas which are resistant to this pathogen, or are we working on a cure for this Fusarium fungus?
So yes, so actually they are large collection of a banana germplast. So I know some of the plant biologists travel around the world to collect different bananas. So they are certain bananas. The pathogen does not kill, at least for today, right? But the problem, we all know that in order for the white type banana or wild banana to taking over the production, it need to taste well. We have to like the look of it, right? It need to stay in the shelf for a certain time. So I remember I had an undergrad course, I would say we are part of the problem too.
Mm.
So actually, one sad story was in Indonesia. So Indonesia has very diverse white bananas. And before, everybody growing their own, and they just cut from their front yard, backyard. But when the industrialization starts, the truck comes, right? The truck comes to harvest the banana. If your banana doesn't look like the same, no one will buy it, which means you will not have business. So therefore, what do? You just chop off your white banana, you're growing the same banana as everybody else. So you sacrifice the diversity in order to make a living. So I would say, every time we say, go to the store, don't buy Cavendish. So you will force the company to, encourage the diversity in the industry. Yeah, so
Yeah.
yes, they are certain bananas are resistant to.
We just have to be willing to expand our repertoire.
The comfort zone, I guess.
Yeah. Well, great. Thank you again so much, Dr. Ma, for being with us tonight. This was really fun conversation, and we look forward to seeing everyone again next month.
Thank you so much for organizing this and for the invitation. And thanks for everyone stay around. I couldn't see who you are, and but thank you so much for coming. And as Allison said, you can post a question to the website. You're probably able to find us too if you wanted to. And so we'll be happy to answer all the remaining questions.
Excellent.
Thank you.